Okay, so now we have three, let's say, industrial talks. So the next talk is for Journal from Zegami about visualization of large data.
Cool. Well, thanks everyone. Especially thanks for having me. My name is Roger Noble. I'm from Zegami. We're a software startup that formed just over 18 months ago out of the University of Oxford. Originally, we were working with the Weather Institute of Molecular Medicine up at the John Radcliffe Hospital, specifically within the Computational Biology group. And they had a lot of problems around the way that they handled huge amounts of images. And so we sort of developed a tool to help them manage all of that. And then since then we've found a whole range of other applications for it, some of which I'll demonstrate today. So to kind of summarize what we're about, what Zegami is, we make information more visual and accessible through intuitive exploration, search and discovery. By combining the power of machine learning and other automated techniques with the power of the human mind, that we can already innately spot patterns and sort of correlations between large data sets. More or less though, we're a visualization tool, and we can view hundreds of thousands of images or any other kind of data points within a single field of view. And then also we also have an interface that kind of makes exploring those data sets really simple and easy to use. The original use cases, as I sort of said, were around high content screening. So these are sort of automated microscopes effectively that take loads and loads of images of lots of cells. And also high throughput plant phenotyping, which is sort of the opposite end, where instead of a small cell, we're imaging tens of thousands of plants within really huge greenhouses. And they're easily generating terabytes of images a day of these things, and they need a way to just get a sense of all of them, to manage them, and also to publish them, to make them accessible to anyone else, rather than just giving a huge big zip file upload somewhere on the internet. We provide an interface that anyone can come and look at the results and look at the source data. Some of the other stuff that I'm going to get to sort of towards the end today is how you can use Zegami as a way to build sort of large ground truth data sets, especially if you're doing any kind of deep learning. That's a really time consuming and expensive thing to be able to do, and we think Zegami makes that a huge amount easier than existing tools. And then also around sort of validating and understanding sort of machine learning models and sort of the outputs of those, so that you can refine them. And then just general analytics as well. You can put any kind of data into Zegami and start to get sort of meaning and results out of them. So it's just so much easier if I show you. So I've got a whole bunch of videos. I didn't want to rely on the internet here to do any kind of live demos. But down the bottom there we've got a URL, and most of these examples that I'll be showing you can look at for yourself on our website. So I'll just jump straight into it. So what you're looking at here is our main interface, and this is an example of a whole bunch of Xenopus embryo. So Xenopus is a frog, and they're taking it at various stages of growth, and we're able to put in this case it's around 300 or so images of these different embryo. Let's check that's playing. From there though, what we're able to do is zoom in and have a look at these images a lot closer. I think my video is playing. Sorry about this. There we go. Okay, so we can zoom in. We can look at any of these particular images. We can also see the metadata that's associated with those. So various different stages of growth. In this case, they were using a different concentration of what's called a morpholino to knock out various sort of gene traits. We can also filter on the metadata we've collected in aggregate. So in this case, what we've then done is sort of filtered down to a particular gene that we're interested in. And what I'll just jump back here because this went too fast. By plotting these against each other in a histogram here, looking at the various concentrations for this particular gene, already we can see there's a visual difference between the ones on the right here and the ones on the left.
What are we actually looking at?
So what we're looking at is a xenopus, which is a frog embryo.
Yeah.
And
you mean a tadpole embryo?
Right, a tadpole. Yep.
Yeah.
Yep. There's a particular looking at expressions of a particular gene, and so there's sort of this dark stain or a marker that's been applied to the embryo, and then the experiment was to look at the concentrations of a substance called a morpholino to see how well or how effective it is at knocking out that gene. So what you should see is if it's doing its job and knocking out the gene, you'll see less dark patterns, whereas if it's not working, you'll see that the stain is still there, if that kind of makes sense. And so what we're able to do in Zegami then is take the images, split them up by the concentrations and then put them against each other, and then you can easily see the differences. And this is just one example of how you can use Zegami as a way to visually compare images like this, and also as a quality control tool. So in this instance, a pair of tweezers got in the way of this one particular xenopus here, and that's just going to muck up your experiment. And no matter how much, I guess, automated image analysis you've done, you're always going to come unstuck with these kind of errors. And again, looking at it in this way makes it, using Zegami, makes it really quick to identify these kinds of data quality issues. The next example that I've got is from the Ashmolean Museum, which is in Oxford. And we have a whole bunch of different artworks that we've done various types of image processing over them. So again, we can zoom in and look at all of the images. We have all the metadata on the side here, but we've also run each of these through an automated feature extraction algorithm to identify various objects within each one of the images. So this is a completely automated way of identifying various things, and then we can use those to filter and explore that data set. This is especially useful if you've got really large collections of images that may have zero metadata associated with them at all. You can really quickly pass them through something like this object extraction, and then use that to sort of refine and sort of start to add metadata where none may have existed. We've also, with this one, extracted the foreground color information as red, green, or blue pixels, and looked at the intensities of those. And then we can in one of our filters plot. In this case, we're plotting the foreground red versus the foreground blue colors into this scatter plot, and then we can use that to then filter the collection. So what I'm just about to do here is circle this bottom region here where there's a lot of foreground red, and you can then very quickly see that it pulls back all the images that have a lot of red in them in the foreground. And similarly, we can do the same thing by circling this other area within our scatter plot, and these ones are predominantly blue. Again, this is really interesting if you're maybe curating a collection of paintings. You can use this to sort of come up with different themes and whatnot. But again, where this is...
What is the method of distinguishing between foreground and background on a painting?
Well, we're just looking at pixel intensity counts.
Yeah, but you mentioned foreground and background, and you're looking at foreground colour.
Yep.
So how do you distinguish foreground from background in your image analysis?
I'm not sure in this case how we did that. Yeah, it may have been that we just looked at the central region and extracted an average colour value of those and then differentiated that with the outer region. I don't remember exactly, to be honest. Yeah. But what I'm doing here in this example is, again, we also looked at, we extracted the sort of the saturation, average saturation of each one of the images, and then filtered down to look at ones that are unsaturated. I'm going to jump back again. What you can see is that, again, that's highlighted a data quality issue where we've got these images here that are missing. They're kind of these grayscale ones. And so we can then really quickly select those within Zegami, add a tag to that, and then we can export that subset of missing images out for sort of to fix them up later on. I've got another interesting one here. This is from a collection of insects from the South Australian Museum. They published a whole lot of their sort of insect collection. And in the same way, again, because we know the location of where each one was collected, we can put together a similar type scatter plot and then use that to find different insects at different locations. We can then view where they're all collected on a larger map. But also, this sort of really highlights, I guess, how Zegami can be extended in a whole lot of different ways. So what we're mostly interested in is having a platform with an API that allows our visualization to be extended and incorporated into other image processing pipelines. And so in this example here, what we did was we extracted a bunch of features from each of the images and indexed those so that we could produce a similarity search. So just previously what had happened was that we were able to select one insect, do a visual similarity to find another 10 insects that look similar, and then from those we can then annotate each one to build up sort of like a training data set. So for example, we could highlight around the front end of this insect and say, well, this is the head or this is the eyes. We can then take all those annotations and start to do some machine learning over that to start classifying, each of these insects. So it's a really highly visual way you can start to annotate and build up data sets for training. The final example I've got is that it doesn't actually have to be images that we're putting into this. It could be anything. And this is just a bit of a made-up example of a whole bunch of financial data related to companies. But you can see here that the colours represent what I'm calling market segment. And then within that, we can have different regions, and the regions can be highlighted by sort of a border around them. And then as you zoom in, we can show additional detail. So we can have this concept of additional sort of varying levels of detail as you zoom in and out of each one of these data points. So this could very easily be sensor data, financial data, or any other kind of data. It doesn't have to be visual, or it doesn't have to be image-based. But again, Zegami provides a nice and intuitive interface to kind of understand the data set from a high level all the way into the discrete individual items. So sort of the quick summary of what I sort of had demonstrated there is that Zegami is an intuitive way to look at really large collections of images. Sort of borrowing from the literary term, it allows you to do a distant reading or look at sort of the aggregate of the entire data set, and then zoom all the way into discrete items in a seamless way. It also is a publishing platform, so you can put this on the web, you can look at it on your phone or on any device and make your data more accessible instead of trapping it in a silo where no one else can see it, or they have to download some certain package or compile X, Y, Z to be able to see your results. And then what we are really building, as I sort of mentioned, is this API so that there's multiple points of extension within Zegami. So each one of these blocks we have an API for that you can develop your own plugins, whether it's sort of on the front end visualizations, but also on the back end that you can plug in your existing processes and pipelines into Zegami, and then Zegami can be that sort of visual layer that sits on top of that all. So to sort of refocus back on maybe the topic of the day, which is that what are the challenges within machine learning and image processing at the moment? One of them is, as with dealing with any kind of data, is data quality issues. And then leading on from that is just understanding the data that you're collecting. I sort of mentioned previous as well that creating these sort of big ground truth data sets, especially if you're doing any kind of deep learning where you need hundreds of thousands of images or data points in order to get any value out of it. Creating those ground truth data sets is really expensive. It's really time consuming to be able to do. And so we really believe that Zegami can make that process a whole lot easier, as well as making it a whole lot easier to sort of get your head around and understand the data sets that you're dealing with. And the other one is that once you have a trained model, is sort of how do you identify, if you are getting incorrect classifications. And so I've got a couple of examples of that that we were playing around with. One of them is of a bunch of hearts that we're doing, working with some of the researchers up at the John Radcliffe Hospital in Oxford, where we're looking at using unsupervised machine learning techniques to sort of compare, diseased and control states, and to see how effective that unsupervised learning is. And then another one, I wanted to compare various pre-trained machine learning, sort of deep learning models, just to see how well they go on a data set that they weren't kind of meant for. So the first example is of the hearts, and in this might be a bit cryptic, but what they've done is they've imaged the hearts and then kind of squished them flat into this circle. And it's a bit like a topographical map, in that this point here in the centre is sort of the pointy bit, and then they're kind of squished into the circle, and you can see there's sort of various markings for the positionings here. We then did a PCA, so principal components analysis, over these images to sort of reduce the number of dimensions down to 2, which we called X and Y. And then we're able to plot those X and Y dimensions against each other to produce this scatter plot. And already you can kind of see there's sort of these two distinct-ish groups. It's a bit faint, but there's sort of a bit of a blob up on the right and a bit more of a kind of a blob on the left. But because these were tagged, these are known hearts, we can then put a series or colour these points to see that our control group is this group over here, and our diseased group is the rest of it. So just by doing some unsupervised machine learning, it's been able to differentiate quite well the two states there. And then we can sort of circle around those items within the UI and sort of add tags and do whatever we like with them. That's sort of just one example of how that's sort of useful for evaluating that kind of analysis. The second one is an Egyptology collection from the National Museum of Antiquities, which is based in the Netherlands. They're associated with the Rijksmuseum. And I was really interested in looking at doing automated feature extraction using various deep learning, sort of pre-trained deep learning models. The first one I wanted to look at was ResNet 50, which is a sort of a 50-layer deep convolutional network. And on the final layer of that, I extracted a sort of a feature vector that I then did a TSNE dimensionality reduction again to reduce it down to sort of an X and a Y. And we can plot them against each other, and you can already see in my scatter plot here, these sort of groupings and clusterings, that if you circle around them or lasso them, that items tend to more or less look the same, even though the model was trained on ImageNet, which is just of cats and dogs and things like that, not of things from Egypt. But again, as useful as that is, you can see in various places, like this example here, where the actual image doesn't match up with the rest of them. So again, it's really fast at identifying these sort of classification problems. But then I also wanted to compare that pre-trained model, ResNet 50, with this another one, Inception V3, to see if that gave us any other benefits. In which case, it's a bit hard to see, but in this example here, the clustering is sort of not as distinct. The groupings aren't as clear, and so even though one model may take longer to process, you can sort of really quickly see that actually the results are a whole lot better. So just some two sort of interesting examples there of where Zegami is sort of useful. So the quick summary is that Zegami it's a unique visualization tool for viewing hundreds of thousands of data points. We're building in a platform, and we're looking for more and more examples, sort of inside of academia and outside of academia, of integrating into existing processes, or sort of working to make life a lot easier when dealing with huge collections of images or other types of data. We actually have many other customers outside of academia and machine learning in particular. One of them is sports analytics, so helping sporting teams pick their players when they're coming up to the draft season, which is really weird for me, but that they love it, which is great. And then, as the other examples sort of showed, sort of digital cultural heritage, so looking at paintings and artworks and that kind of thing as a way that you can provide an analytical interface for these types of institutions, so that people from around the world can not only get access to them, but then start to get value out of them, not just looking at one or 10 things at a time, but they can see 10,000 things at a time, which is really cool. That's it. Thanks. Time for questions?
Just time for one question.
Yeah.
Can it recognise handwriting on old manuscripts?
So that's sort of part of what we can build into that pipeline. So it could be used to sort of display the results of some handwriting recognition or analysis, if that kind of makes sense. So you can sort of plug in a handwriting recognition function.
So you take the output from that and then put it in.
Correct. Yeah.
So it could be helpful, therefore, in identifying identical authorship of different manuscripts.
Yeah, that's right. Yeah, yeah. Or just identifying, if your OCR algorithm got it right or not. So it's a validation tool.
Yeah.